Great. And Srini Turaga, would you introduce yourself in 30 seconds or so? Yes, unmute
Great.
yourself.
Yeah, thank you. Yeah. So I did my PhD at MIT under Sebastian Sung, and at the time he was working on computational neuroscience modeling, and then during my PhD decided to change focus completely, and he said, well, what can we do with electron microscopy? Can we map the structure of real neural networks in the brain? And that sort of started a new field called connectomics, where people are now doing 3D electron microscopy to map individual connections in real nervous systems to see what do real nervous systems look like. So we developed at the time convolutional neural networks to help solve the mapping problem, and then the field became called deep learning. And now that we have connectomes, my lab at Janelia at the Howard Hughes Medical Institute works on interpreting connectomes and building computational models of real living neural networks in the brain using constraints that we get from the structure of real neural networks as well as functional measurements and so on. We also work on computational microscopy techniques and protein engineering. So a bunch of places where you can use deep learning to analyze data as well as to build models.
Super cool. And Jakob, you want to introduce yourself?
Hi, my name is Jakob Macke. I'm a professor for machine learning in the sciences in Tübingen University. I could never decide whether I want to be a machine learning researcher or computational neuroscientist. So basically from day one of my research career, I've been doing both. And particularly we've developing machine learning methods for neurodata analysis. And for some of the work, we realized that when you want to fit models to data, then you run into inference problems that are very hard to solve with traditional means. So we've in particular been using neural networks as tools to allow us to fit models to data more efficiently. And some of the work, in fact, is in collaboration with Srini, and particularly the work on where we together are trying to kind of use these kind of methods to get better signal out of microscopy data.
Fantastic. All right, let's get to some questions because we have a short session today. So there's today's theme was it's the second day of deep learning, and today we looked at autoencoders and variational autoencoders. And so we want to try to make some connections and understand what they not just can do practically, but what they might tell us about computations and maybe about something that might be going on in the brain. So Cedric Foucault here is asking, are there any speculations about how autoencoders could allow us to efficiently store and replay episodic memory? And to what extent might these ideas be supported by any empirical evidence? So I don't know, does somebody want to jump in and start? Tim, I feel like you might know something about this.
Sure, yeah, I know something about it. I mean, so this is, I think, going to be an extremely interesting question going forward. I can tell you very quickly what's happening on this front in the deep learning case, which I think is ultimately kind of unsatisfying in the context of deep reinforcement learning, which I think is ultimately kind of unsatisfying as far as what we might want to do, what might be going on in biology and what we might want to do in future. I mean, at the moment, people are still, in effect, just storing raw inputs when they build the replay buffers. So raw images, other raw sensory modalities, and the rewards that have been collected along the way. And although there's been a lot of speculation that maybe what you could store is sort of like some encoding of the environment into replay buffers and use that when you're doing off-policy learning. I don't know of any notable cases where people really have that working well. So that is, they basically, the evidence so far is that if you can store the raw stuff and like re-encode with whatever networks you have at the moment when you do your off-policy learning, that just seems to work better. So with all the algorithms that we have. Now, there's some interesting questions there. I mean, one of them is maybe, you know, so the brain doesn't have this luxury potentially in terms of like raw storage capacity. So that might be part of what's going on. There's also speculations. People also speculate that, you know, if we really had the right ways of doing these encodings, you could do these kinds of off-policy algorithms much better. And this is kind of the vision or dream of that, which would be as follows. So if I've stored a bunch of encodings in these sort of like a bunch of vectors in this encoded space, where similarity metrics are much more interesting, then instead of doing replay along a sequence, which was just the raw sequence that I saw in the environment when I acted in it, then I could do replays along sequences which are a mix of sort of stuff that I actually saw, and then transitions into other trajectories that I haven't seen, because of sort of something like a similarity of states. And so potentially, if you could make this work, you could start doing off-policy replay along, you know, putting together pieces of sequence that you'd seen in the real world. So far, I don't know of algorithms that do this effectively. I think it's probably one of the coolest things to look at if people are interested algorithm-wise. Yeah,
that's very cool. So I mean, I guess one thing that we can think about is just in general, in order to encode memories efficiently, we have to do a lot of compression, right? And if you can compress it into a space which has, as we do in, say, a variational autoencoder, some kind of topology and structure that may be useful for doing inference and for reproduction, that's already actually, you know, at least a first order way of thinking about it. I'm just thinking here of an example of when we do visual recall, we have to, you know, sort of use our generative model to push back what we think we were thinking about into sensory cortices, or, you know, specifically visual cortices if it's visual imagery. And, you know, if you think about an autoencoder as something that generates that low dimensional space, which could be used for memory, but then also, you know, has a mapping back to that original space, you know, it might give us some insights into, you know, how we actually efficiently can reconstruct those higher dimensional memories from some smaller, lower dimensional space. Okay, so let's move on because we don't have a lot of time. So we have here a basic question. What are the strengths and weaknesses of autoencoders compared to other dimensionality reduction techniques? I don't know. Jakob, you want to give that a stab?
I can have a shot at that. So, so autoencoders and variational autoencoders, they, rather than being like a specific algorithm, they really are a framework for building algorithms. So in that sense, this is both the strength and the weakness of this approach. The strength is that kind of in this autoencoder framework, we can build a variety of different algorithms that are suited to different analysis tasks, or that incorporate different amount of knowledge that we might have about a task. So the generality is one strength. The second strength is that it explicitly includes a generative model of the data. So some dimensionality reduction algorithms allows you to reduce the dimensionality, but then you can't go back from the low dimensional space to the high dimensional space, at least not in a principled manner. Whereas for autoencoders, you can change something in latent space and see what's the effect of that in my data space. So it's really a particular form of generative model. And in terms of weaknesses, one weakness is the generality. So you have to make decisions on what to build. The other weakness, in particular for variational autoencoders, that there's some very subtle and some not so subtle optimization questions that can come into play that can really affect their performance. This is a question that I think goes more into machine learning questions rather than neuroscience questions, but trying to get a variational autoencoder work well on a specific task, if they're complex tasks, you can run into all sorts of optimization problems.
That's great. So let's see. I guess we've sort of already touched on this, but there's a couple of related questions, which I'll just throw out to the table. So the related questions are, does the brain really have anything like a convolutional filter? We're using these kinds of convolutional networks, and that operation, is it really biologically plausible? And then sort of another question is, actually, is there any evidence that the brain can do gradient descent? And another related question is, do we have any evidence for actually seeing something like an autoencoder in the brain. So I don't know. Srini, do you want to take a stab at that?
Sure. Yeah.
Any one of them.
So, yeah. I'll start with the convolutional network because that's something that we're kind of working on right now. Well, the convolutional networks have two components to them. One is local receptive fields, and what that means is each pixel in a feature map looks at a local neighborhood of pixels in the previous feature map coming in or in the input space. Now, retina and both in mammalian retinas as well as the fruit fly retina, which I work with now, certainly has that. And visual cortex as well, we know that there's localized receptive fields, meaning that neurons only, they're not fully connected. Neurons in one layer don't receive input from all the neurons in the previous layer, just from a local subset of these neurons. And so that's awesome. But the place where this sort of breaks down is convolutional networks also say that the filter in this place, the weights in this part of the feature map, and the weights in this other part of the feature map or visual space are exactly the same. And so that's clearly an idealization. In biology, you never see that exactly because these are two different neurons that wired up independently. And so, there's always some differences there, even though they might actually be trying to have the same weights, either through experience or some form of innate sort of programming. There can be some differences. But let's say in the fruit fly visual system that I'm looking at, they're very close to the same. So an idealization of treating them as exactly the same is sort of reasonable. That's one of the questions. I forget, there are three others. I'll take one on, is there variational autoencoders in the brain? And I'll turn it around a little bit to talk about something that that I think is very relevant. When we have models of real nervous systems that, let's say, we're trying to build, in our case, what we might have is the decoder part of the autoencoder might actually be a model of the visual system, let's say, or a model of the nervous system, the entire nervous system of a C. elegans worm. And that's generating all the things that we're measuring. And then you might use the encoder part just to do inference in this model so that you can actually fit this model. And so this is a way of basically bringing biology and grounding it on one part of the autoencoder, the decoder part, and then the encoder part is very general. And so, this is another place where you can ask the question: are autoencoders interpretable? This is one way of making them interpretable, because the latent variables are activities of individual neurons that you measure.
That's great. So I think the other questions, so there was a question basically about whether feedback alignment, and I sort of generalized it a bit. When we train these deep convolutional neural networks or any deep network, we use normally stochastic gradient descent and its friends with lots of funky engineering tricks whenever we can. But is there sort of plausible ideas about how this is being done in the brain? Does sort of directed feedback, and we know that there's feed forward and feedback connections across cortex. Is there evidence that in fact the brain is doing something like this? Is this just a completely open question to which we don't know the answer? Would someone like to speculate?
So,
oh, sorry, Marco. Let's also say, Marco, just give a few words. This is our content creator for the day, so he's to be praised. And I guess I'm to be blamed because I helped him organize his thoughts. So Marco, just give yourself a 20-second introduction and then go ahead.
Hi, everyone. So I'm working in industry in a small startup for automated document recognition, and I've done research in neuroscience, computational, and sleep research. And I love the summer school, and I've been doing this for a few years. And it's really great that now we have this global one online that everybody can participate and benefit from content. It's really great to participate in this project.
Was that quick enough? All right. So regarding gradient descent, if we look at it in terms of learning from mistakes, right? So we have our gradient descent because something didn't match up in our optimization function. So we have some error signal, and this error signal is used to update something, right? So I guess some process like this, we definitely have something. Well, I would speculate that the brain in some sense has a mechanism like this. And jumping back to the previous question, whereas we have autoencoders or not in the brain, there's an application where you have, so you have video scenes, and the autoencoder is learning from the past video scenes and tries to predict a new one. And if we think this about our internal model of the world, we are constantly having this feedback, these signals that come from the outside world, and some sort of autoencoder could be on the lookout, could be like having a prediction of what comes next. And whenever what comes next is not the expected thing, right? So we detect that something is not as expected, and then we can attend to it. So I would see autoencoders, yeah, something like this.
Yeah, and I guess, following up on what you said, we're very aware that the brain calculates the sort of partial component of what we need to see for backpropagation in some sense, which is that we see predictive error signals that would be sort of let's call it the simple scalar part of the error term. And the question is how we could divide that up, right, across neurons to really allocate that error appropriately. And I think that's where things get a little challenging. I don't know, Tim, if you wanted to add a quick thought to that.
Yeah, I think, with respect, I just sort of said in the chat, with respect to feedback alignment, I think it's pretty clear that that sort of algorithm in particular won't scale up to hard problems. We've looked at that now, and that's good. I think of it more as a starting point for, reminding us that the effective approximations to backprop and SGD might actually be quite simple and reasonably easy to implement. There have been a couple with connected to autoencoders. Actually, we talked about in that paper that actually in the feedback alignment paper that if the backward running weight matrices were not random, but actually something like a pseudo inverse, which is basically sort of what you would get in a autoencoder situation, that a pseudo inverse actually will work pretty well for credit assignment in a backprop like setting. So and one can also imagine that learning a sort of autoencoder in the brain might be reasonably easy. And so I think looking down that direction makes a lot of sense. But yeah, I wouldn't get too stuck on feedback alignment as a real scalable alternative.
Well, I think one of the things that this should remind all of us in the audience, well, as I should say, all of us, the panelists and everyone in the audience, is this is great. It means there's work to be done. There's science you can engage in. There's questions you can answer. And that's what we should all be looking for. I'm going to pull a question here, which I think is kind of an interesting one. Maybe it allows a couple of creative answers. So it's from Antonio Falasconi, and it is, do you think we could make sense of the geometry of movements? And I'm assuming he means through using autoencoders. And what do you think the latent variables would be like? What would the structure of that be if we could do it? I don't know. Jakob, do you want to speculate?
Yeah, I can have a shot at that. I'll try to kind of interpret the question even a bit more broadly, and that is that when we look at what the brain does, it controls behavior. So if you want to look at latent structure in the brain, then that will somehow have relationship with the latent structure of behavior. So if you want to really understand what the kind of organizational units of the brain are, part of that answer can come from really looking at what are the underlying structures and behavior. So when we take high-dimensional behavioral data, for example, movements, are there some underlying principles, smaller modules, manifolds that really seem to explain most of the behavior and the movement, and then look for how they would map onto neural activity. So I think there is kind of a lot of interesting research happening that is trying to address these questions by looking at using autoencoders and other techniques to really look at hidden structure or low dimensional structure embedded in behavioral data. This has a long history, I'm starting from ideas such as movement primitives and synergies, but there's very modern versions of this that really rely in a very heavy manner on modern deep learning tools for finding low-dimensional and interpretable, ideally interpretable structure in behavioral data, such as movement tracking data.
Let me just ask a slight, hopefully quick follow-up question, which is, you know, classically in the motor literature, when people started looking for sort of low-dimensional representations in the motor system, I would say they went with what we, the external experimenter, thought might be convenient for us. You know, do we see vectors that correspond to, say, retinotopic space or some simple effector space? And we found that, oh, after looking a little bit harder than the first experiments, it quickly broke down, right? It was a more complicated representation. So I'm curious, do you think that the kinds of those low dimensional manifolds we're finding, are they going to be something that's going to only partially align with our intuitions from the outside as sort of observers on what the representational structure should be? You know, or are they going to be completely uninterpretable in the sense that if we know what they are, we can look inside the brain and maybe we can find those representations being used, but we'll never really understand what the space or structure of them is?
So to some extent, I think it'll depend on the scale at which we're trying to understand behavior. You know, the details of millisecond by millisecond sort of timing of how we control our limbs, that will depend on exactly how our limbs are actuated, where the muscles are, and you know, the details of how the muscle responds and how our limbs are articulated. But at a higher scale, you know, if you think about, did I pick up this mug? Am I drinking it? That level, you know, it doesn't matter what effector you're using. You have to sort of probably send the same commands. So at that scale, you can probably just look at the behavior and try to interpret, you know, try to build models of what sequences of events might have to happen, and try to understand that sort of ordering of neural activity, let's say. And that's probably, you'll see that in the brain somewhere. But at the finer scale, we'll need models of the finer scale details of the body. We'll need an embodied sort of model, and then if we build that into, you know, part of the decoder of our autoencoder, then we'll probably see the right things emerge.
That I think that's something that, I mean, I think that's something that we have to take home from this course overall, right? That is sort of a fundamental property of trying to understand the brain. And this has come up a couple of times in my question and answer sessions. Like we have to choose the level of description for the particular question we're asking in the particular moment. And that means abstracting away some details, focusing on others. But at the end, in order to really understand a system, we're going to have to, you know, have multiple levels of understanding, you know, trading off the kind of fine-grained understanding for you know, say heuristic efficiency and simplicity. And that brings me to what may be our last question, but I think we could play with it a little bit, which is from Matthew Sargent. And it's basically this question. So deep learning abstracts away some of the finer details of neurons. Do spikes have some computational advantage beyond energy consumpting, or are deep learning models really going to be sufficient to explain everything at the end? Is it all rate coding and some form of gradient descent that we don't quite yet understand? So, Tim?
Yeah, I'm happy to weigh in at least briefly. I think this is one of these questions which, from the time of sort of like, I guess, being an undergrad for me to now, I felt like when I was an undergrad, it was a total mystery and really some questions left. And now it feels relatively straightforward. It really does feel like it's going to be ultimately just about energy consumption. We shouldn't knock that. I mean, if you want to have a pretty powerful network in one of your cell phones, then, you know, maybe you'd better get serious about things like spiking and so on, possibly. But as far as I can make out, in terms of can you reach the sort of like human level performance on XYZ, I think the jury is in, as far as I'm concerned. I mean, you can do things like play Go at levels that are superhuman, and then if you haven't checked it out already, go and read the GPT-3 paper in detail and prepare to, you know, be shocked if you haven't already looked at it. You know, go do those things. And I think if you once you've got through those, probably most of your doubts about this should dissolve. Yeah.
Does anyone else have, I don't know, somebody want to take the counter position?
I mean, it's not really a counter position, just sort of adding two components to this. As Tim said, energy might make all the difference, right? Because if you want to scale up further in an autonomous system, then you might need to go to a solution that's radically more energy efficient. And the second point is also, the question left it a little bit ambiguous when we mean computation advantage, whether we mean like for artificial systems. And as Tim showed, artificial systems that don't use spikes can be, I think, vastly more powerful than we ever thought they could be, or at least certainly mean. But the question of how things are implemented at a computational level in the brain is not the same question. And it's not the same question because it's tightly bound to the biological mechanisms that can be used. Part of that is energy efficiency, but that might not be the only one that forces or gives a more fundamental role to spikes. But certainly for artificial systems, and if you take energy consumption, if you leave that out, I think there's overwhelming evidence that deep nets can work much better and can be much more powerful than we thought they would be. Doesn't mean they can do everything. There might be other things where they hit limitations that will certainly, that's already evident now, but certainly they can be more powerful than we ever thought they could.
I think that's a good way of saying it. I'll get to you in 1 second. So they're much more powerful than we thought. But say the AlphaGo or one of these systems that's an absolute expert, right, in a domain will beat the best human grandmaster, will still lose at being a generalist to your average 2-year-old. So that's not about spiking versus not spiking. It's to say there's a long way to go to understand the trade-offs that evolution has made to allow us to have both strengths in our intelligence and our capacities to learn and to compute, but also to be able to handle all kinds of crazy situations. I'm just gonna drop in the thought that if you look and say the auditory system, spike timing is actually quite important for perceiving information. And I'll bet you if we looked through the brain, there's going to be other places where for domain specific reasons or computation specific reasons, whether those spikes are there for energetic reasons or not, they're being used. And maybe in the same way, there's probably other properties, be it oscillations or LFPs that can do coupling locally. There may be a bunch of other ways in which we don't know what the cause or effect was, but once the system has a tool, it can take advantage of it. And so I'm betting that we will find that there will be some diversity in how we're going to have to think about the kinds of things that underlie the specific computation. So sorry, Srini, please.
Yeah, this is actually particularly relevant to what you're saying. So I'll just add some facts. Invertebrate nervous systems, like C. elegans, this is a worm that just has 302 neurons. So it presumably is very efficient with the neurons that it does have. Most of those neurons don't spike. The fruit fly visual system, which again, the fruit fly has very few neurons. It's a tiny little animal and a tiny nervous system. Most of its neurons also do not spike. So that's on one side of things. Now, there are neurons in all of these nervous systems that do spike, and some that are partly non-spiking and then partly sometimes do spike. So what is the role of that? I don't know. Now on the artificial side, we typically try to train neural networks with continuous activation functions, but of course on the computer everything's implemented discretely. And frequently what people try to do is to quantize these networks so that they use less memory. Quantization, you can ask, it's not exactly the same as spiking, but it certainly reduces the dynamic range of the activity variables that you can imagine having in your model. And that's more energy efficient. So even in artificial systems, we try to do this. So I don't know the answers, but here are some facts about what we know, and I think it's food for thought.
So I'm going to give Marco the last word because he made the tutorials for today. So Marco, I know you've done some work on spiking systems. So why don't you close out?
Well, on my closeout, actually, I would like to mention something else, is that we have not seen these deep learning systems interacting freely with their environment, right? And we're comparing what our humans and our neurons are doing, but it's a totally different. We're just giving them very specific problems. But I think pretty soon we'll have them interacting, and they have one advantage over us, is like, how much information am I conveying in these 30 seconds I'm talking, and how much copy and paste can these systems do between them, right? So there are some advantages.
Yeah, I mean, I think it's always wise to remember that a few 100 million years of evolution probably has managed to solve some problems in an efficient way, and that we'll be able to learn from it. It doesn't mean that we won't be able to cleverly outdo that in particular problem domains and eventually, achieve something similar. But it should be, I mean, I like to think, so since I do RL, I like to think that the solution that was found at some level to sort of figure out what's the best thing to do is the same in the honeybee as it is in us. So there was something pretty good about that solution because it's been around for a really long time. And actually a lot of things that we look at in brains are quite similar. So evolution does sometimes go down a garden path, but there's probably a reason to believe that it's not entirely a crazy architecture to do intelligent things in an embodied context.
And with that, problems.
Yeah, we have to let this wonderful panelists go. So thank you all. It's been a great pleasure. Sorry this was a shortened session, but we have to let the students do their evaluations of the course, which we really, really, really need. We need to know what we did well, what we did badly. This is a complete scientific experiment that we have just conducted. 3-week interactive online summer school, basically pulled together in 3 months with the amazing support of the entire community. We really appreciate it, and we really need to know what we can do better next time, what we did well this time. And so I see we have the next generation of young scientists here with us. Thank you all. Thank you, Tim. Thank you, Marco. Thank you, Srini. Thank you, Jakob. Look forward to chatting soon.
Thanks, everyone. This was fantastic. Thanks all.
Everyone.
Thank you. Bye.
And thank you all the students.